So as I promised, I'm going to record this little fragment here about the um, respiration, the part that didn't record it well. So we briefly spoke about physical factors that influence the ventilation, right? And we talked about the airway resistance. And we figured that airway resistance is not really a significant factor. It's rather insignificant. However, it is still, the higher the resistance, the lower the flow is going to be. Basically, the airflow, as it is shown in this equation here, is airflow is directly proportional to the pressure and inversely proportional to the resistance, meaning the higher the pressure gradient, the higher the flow, the higher the resistance, the lower the flow. So medium-sized bronchi, they show the maximum resistance. The reasons, so if you would compare it to the blood flow, resistance is highest in the small arterioles, right? Now, why it's not highest in the terminal bronchioles? The reason for it is that in the terminal bronchioles, the diffusion happens. So air starts to diffuse through the walls of bronchioles, and resistance essentially drops to almost zero. And of course, the diseases that would affect that part of pulmonary ventilation, that factor, would be any type of like asthma or suffocation, any other type of a condition that would constrict bronchiole or obstruct it with the mucus. As we mentioned, surface tension. Surface tension plays a critical role in the maintenance of the alveolar shape. And I want to remind you that basically surface tension is the force with which air molecules of water, shown here, it's a molecule of water, attract to each other. When alveolus inflates and enlarges, surface tension of the fluid that covers the inside surface of the alveolus, surface tension of that fluid increases because water molecules, they are separated, basically like a balloon. When you inflate it, the tension of the balloon surface increases. Same happens here. As you inflate alveolus, the tension of the fluid inside the alveolus increases. Now, this force, this surface tension, this water surface tension that tries to basically collapse the alveolus, acts against the inflation. Surfactant that consists of lipids and proteins produced by type 2 alveolar cell, surfactant decreases the surface tension of the alveolar fluid, which means that these molecules will attract to each other with the lower force and will less likely cause the collapse of alveolus, basically. For instance, in premature infants, when they do not produce sufficient amount of surfactant, what can happen after a breath, alveoli can collapse. And to inflate them, they have to, babies have to apply much greater effort because now surface tension is not reduced and works essentially against the inspiratory effort. Now, as we mentioned, lung compliance is the degree to which lungs increase their volume with each change of transpulmonary pressure. Now, I want to remind you that if this is the lung and this is the pleural cavity, then transpulmonary pressure is essentially between the lung and the pulmonary cavity, okay? Pleural cavity, I mean. If we go back several steps, so that's transpulmonary pressure shown here. Okay, and you can see that transpulmonary pressure is the highest at the difference is the highest at the peak of inspiration. Now, the more lungs inflate at certain transpulmonary pressure, the higher the lung compliance, which basically means that the higher compliance, lungs are really easy to inflate. The inspiration is easy, but expiration is extremely difficult. The lower compliance, lungs are really hard to inflate. Inhalation is hard, but expiration is really easy. And essentially, the formula for the lung compliance is shown here in the right bottom corner. So you see, it's the change in the lung volume that corresponds to a certain change in the transpulmonary pressure. So what affects lung compliance? Mostly the structure of the lung stroma itself. Scar tissue that replaces the lung tissue during fibrosis. Of course, low production of surfactant, and thoracic cage, lack of flexibility. For instance, when people lean forward, when they have kyphosis, that reduces the lung compliance. Ossification of the cartilage that connects ribs to the sternum reduces lung compliance, and intercostal muscle paralysis. Now, this four volumes, four capacities shown here, and two capacities shown on the next slide, you must know them all. What these volumes are. I drew the picture for you, and basically what we try to show here is that you have a lung, right? So that's going to be a tidal volume. Now, the volume that you can inspire on top of the tidal volume is going to be inspiratory reserve volume. Volume that you can expire on top of the tidal volume is expiratory reserve volume. And this, the volume that you can never get out of the lungs, because otherwise lungs would collapse. Volume that is left in the lungs after a complete expiratory effort is called residual volume. Now, speaking of the capacities, what happens here, again, if this is the lung, and this is the tidal volume, and that's inspiratory reserve volume, expiratory reserve volume. Of course, the total volume is fairly easy, right? Total capacity, sorry. Vital capacity is everything except residual volume. Okay? Inspiratory capacity, tidal volume plus inspiratory reserve volume. Let me correct myself here. Okay. So that's tidal volume, that's inspiratory reserve volume. This is expiratory reserve volume. All right. So, sorry about that. So essentially, and functional residual capacity here is the expiratory reserve volume plus residual volume. So know these capacities. There will be questions on the exam. Probably saw them in the quiz already. Get ready for them. Dead space. Anatomical dead space represents the volume of the respiratory passages. Essentially, air that stays in a bronchi, trachea, bronchioles, pharynx, larynx does not contribute to gas exchange. It's about 150 milliliters. That's why it's called dead space. Shown here, it's the beige respiratory passages. And then you have alveolar dead space, alveoli that do not participate in gas exchange. Problems with the circulation, obstruction of the alveoli themselves. So altogether, this anatomical dead space and alveolar dead space, they comprise total dead space. And we can essentially measure the respiratory volumes using a device called spirometer, which allows us to actually distinguish between two types of the respiratory disease: obstructive, in which respiratory passages are blocked somehow, say by mucus in case of bronchitis, or restrictive, which is a type of disease in which surface area of alveoli is decreased or functional volume, total lung volume is reduced. Total lung capacity is reduced, let's say. So how can we figure it out? Think about this. We can measure how fast the person can expire air, and what is the forced vital capacity, how much air the person can expire at max effort. So in obstructive condition, the total amount of air that is in the lungs doesn't really change, so forced vital capacity remains basically normal. However, the ability to expire it at certain rate decreases, so forced expiratory volume, amount of air that is expelled during, say, 1 minute or 1 second, reduces, because person has to expire through the smaller respiratory passages, so the flow, the rate goes down. Now, in restrictive disease, forced vital capacity goes down because basically the functional capacities of the lung, they reduced. However, the rate of flow is essentially the same. Now, minute ventilation is the amount of air that reaches the lungs in 1 minute, essentially the volume of one breath, the tidal volume, multiplied by the respiratory rate. You can see it here in this example. It's 10,000 mils per minute. Now, the problem is that you still have dead space, and dead space does not contribute to actually gas exchange. So to see how much air reaches alveoli, we calculate alveolar ventilation rate, which is tidal volume minus dead space multiplied by the frequency of breathing. So here in this example, you can see that in case of slow and deep breathing, when you take in a lot of air with each breath, although your breathing rate is fairly slow, the amount of air that reaches alveoli with each breath, here in this example, second line, see second line here. The amount of air that reaches the alveoli are 850 mils with each breath. So effective alveolar ventilation is 8,500. And a ratio between the alveolar ventilation rate and minute ventilation rate is 85%, 15% more than at rest, normal ventilation. Rapid shallow breathing, dead space doesn't change, but tidal volume actually decreases to 250. So alveolar ventilation rate drops precipitously to 4,000 milliliters per minute, which makes it 30% less effective than normal breathing. For the exam, you have to be able to calculate minute ventilation and or alveolar ventilation based on the data that I give you. Non-respiratory air movements. You just, got to know which ones are non-respiratory air movements, what triggers them. Well, except basically hiccups and yawn, because we still have no idea what triggers them. Know for hiccups that it's a diaphragm spasm. Know for cough and sneeze that those are irritants being in the respiratory tract. crying and laughing essentially are the same, just different emotions usually that trigger them. And the function of cough and sneeze is to clear the respiratory passages, the gas exchange. So I have two respirations here. We have external and we have internal. In external respiration, carbon dioxide here goes from blood to the alveoli and eventually is exhaled. And oxygen goes from alveoli to blood and essentially is inhaled. And in internal respiration, carbon dioxide goes from tissues to the blood and oxygen goes from blood to tissues. So what drives this gas diffusion? It's the difference in partial pressures of gases between tissue, blood, and alveoli. So to understand this, we need to define the partial pressure, PI, partial pressure. It's the pressure of each individual gas in a mixture of gases. And the Dalton's law says that the total pressure of the gas mixture is the sum of partial pressures of gases that compose that mixture. And the partial pressure is proportionate to the basically the concentration of gas in the mixture, the share of it. Henry's law suggests that concentration of gas in the water is proportional to its partial pressure, which means that the pressure at the equilibrium pressure in the liquid phase and in the air, pressures are the same for the same gas. So it actually allows us to use partial pressure as the measure of concentration of gas in the blood, because they are in the equilibrium here. One important thing is that temperature, higher temperature makes all gases less soluble in water and vice versa. So that's the composition of alveolar gas. And you see that there's very little CO2, very little water. But as we look in the alveoli, the percentage of water and CO2 dramatically increases. And since we're talking about 100% all the time, means that the percentage of oxygen and nitrogen decreases, which actually means that actual pressure of oxygen in alveoli is lower than the pressure of oxygen in the air. So that's what we actually have in alveoli, okay? So that's the, this, and you can see that what's going on now? You can see that as the pressure of CO2 increases, pressure of oxygen decreases because it still has to be the same pressure, 760 millimeters of mercury. So if one pressure in the mixture goes up, other pressure has to go down. Now, the exchange of oxygen and CO2 happens across fairly thin respiratory membrane, which consists of the endothelial cells in the capillary, type 1 alveolar cells in the alveolus, and the basement membrane here, yellow in between. Okay. So this membrane is rather thin. It is thin because of those really tiny little layers, and it has enormous surface area, about 60 square meters. Now, if lungs become filled with water, like there's pulmonary edema or pneumonia, which leads to accumulation of water in the lungs, gas exchange suffers, of course, because membrane becomes thicker. Now, if emphysema and some other restrictive diseases, the surface area of alveoli goes down. which means there's less surface area available for the gas exchange, which means gas exchange suffers from it. Now, in external respiration, oxygen in the alveoli, its partial pressure is 104, and partial pressure of CO2 is 40. You can see these numbers. Meanwhile, the partial pressure of gases in the blood that enters the lungs from right ventricle, oxygen is 40 and CO2 is 45. So you can see that pressure of oxygen in alveoli higher than in the blood, which means oxygen diffuses into the blood. It follows the pressure gradient. However, CO2 in the lungs, the partial pressure of carbon dioxide is lower than in the venous blood, which means CO2 now go from the venous blood to the alveoli. Okay? CO2, since it's more soluble in plasma, it does not need that much of a gradient, that steep gradient, so it's all good. Now so it diffuses very fast as well. This shows you how fast the gas exchange happens. You can see that normally gas exchange requires 1/3 of the time that blood actually flows through the pulmonary capillary. It's really important because in case of exercise, when blood flow dramatically increases in terms of the speed, there is still a lot of time, extra time, for the gas exchange. So even if now the blood flows faster and there's only, say, 0.5 or 0.25 seconds left for the gas exchange, it's still enough. So basically what it comes down to is that there is never a situation when there's not enough time for the gas exchange in the lungs. Now, of course, during the gas exchange, external respiration, the maximum amount of blood should flow to the alveoli with the highest concentration of oxygen. Okay? So when concentration of oxygen in the alveolus increases, blood vessels that lead to this alveolus, arterioles that lead to this alveolus, they dilate, allowing more blood to get to the alveolus with a lot of oxygen, okay, to pick up that oxygen. However, if alveolus has very little oxygen, then blood flow to that particular alveolus will go dramatically down, okay? You can see that lower level of oxygen will lead to the constriction of the arterioles leading to that alveolus, meaning that blood will be redirected to the alveoli with a higher concentration of oxygen. Now, what happens with the ventilation? When there is a alveolus with a lot of CO2, what do we need to do with that extra CO2? We need to exhale it. How can we do it? We dilate bronchioles, we increase the diameter of respiratory passages, so CO2, which is high at that particular alveolus, leads that part of the lung much faster, bringing the oxygen levels up. If CO2 in alveolus is low, bronchioles constrict, allowing oxygen to stay in the alveolus. I hope it makes sense because it allows to exhale CO2 faster, essentially. Now, so we've got oxygen that goes from lungs to the blood, and we've got CO2 that goes from the blood into the alveoli. So essentially, the blood that leaves the lungs and enters the capillaries has a lot of oxygen and very little CO2. Well, in tissues, the concentration of oxygen is fairly low, so blood in tissues. So oxygen leaves the blood and enters the tissues. So oxygen leaves the blood and enters the tissues. While CO2 concentration in tissues is pretty high, so CO2 goes into the blood where the concentration is low. Remember, gas always diffuses from high partial pressure to low partial pressure. Okay, so partial pressure of CO2 in tissues is higher than partial pressure of CO2 in the systemic blood. There you go. CO2 diffuses into the systemic blood. And then this blood now, with low oxygen and high CO2, gets into the alveoli, and the gas exchange happens again. Now, what carries oxygen in the blood? We're going to talk about it. I'm going to wrap it up. It's going to short and sweet presentation. Oxygen in the blood is carried mostly by hemoglobin. So as we discussed before, each molecule of hemoglobin consists of 4 subunits. Each subunit carries a molecule of heme, and each molecule of heme, with its iron in the center, can bind 1 molecule of oxygen. So total, 1 molecule of hemoglobin can carry 4 molecules of oxygen. When hemoglobin binds oxygen, it becomes oxyhemoglobin. When it unloads oxygen, like a truck, okay, becomes deoxyhemoglobin. The number of oxygens that are bound to hemoglobin determine its saturation. If there are 3 oxygens, saturation is 75%. Now, loading and unloading. It follows the cooperative binding rule, which means each next molecule of oxygen is loaded easier. First one is really hard to get on. Okay. Second is easier. Third is even easier. And fourth is absolutely easy. So each consequent molecule of oxygen is easier to bind to the molecule of hemoglobin. That means cooperative binding. When all 4 bound, hemoglobin is fully saturated. We discussed the idea that actually you always have a mixture of molecules in the blood, which means some molecules are going to have 4 oxygens, some hemoglobin is going to have 3, 2, 1, none. So essentially, average of those saturation numbers for each molecule average will show you the actual saturation of hemoglobin in the blood. It can be 80, 81, 82, and whatnot. Now, rate of loading and unloading, it depends greatly on the partial pressure of oxygen in alveoli, temperature in alveolar tissues, blood and tissue pH, partial pressure of CO2, and concentration of the BPG, the molecule that's produced during glycolysis by red blood cells. When oxygen levels are low, levels of BPG dramatically go up because of hypoxia and basically make the loading of hemoglobin much easier. So we're going to wrap up at it because we talked about hemoglobin dissociation curve, and you can ask me any questions in the class, and I will see you tomorrow.